and very good scientist. He is none other than Dr. T. Shivanandappa. He is going to speak on evolution of genes, form and functions. So it is my great pleasure to introduce yourself to you all. Dr. Shivanandappa is one of the prestigious alumni of Zoology department and also University of Mysore. He has completed his MSc PhD from University of Mysore only. Although he was very much interested in teaching field, but he could get golden opportunity to work in one of the prestigious national laboratory, that is CFTRI Mysore, where he served as a scientist and worked up to director level cadre. Dr. Shivanandappa research interests are many. He is a multi-faced personality. He is expertise in reproductive biology, endocrinology, animal physiology, genetics and genomics, wildlife, etc. He has published more than 100 research papers. Most of them are having very high impact factors. He has attended various conferences and also conducted various conferences at the national and international level. He has visited several countries. And he has completed 6 projects. His citation index is 1265 and H index is 28. I-10 index is more than 55. He has guided more than 10 PhD students from CFTRI as well as from Zoology Department, University of Mysore. With this brief introduction, I welcome you, sir, and over to Dr. Shivanandappa, sir. Thank you, sir.
Thank you. Is it directly, Alva? Thank you, Professor Basurajappa.
Thank you, sir. Thank you very much.
Okay. So, good morning to all the participants of this meeting. Am I audible?
Yes, sir.
Oh,
Yes, sir.
okay. I'm just switching my phone off because it's a problem. Oh, okay. So let me start with the topic I have chosen. I guess among the participants, we have probably mainly biology group, life sciences, biochemistry, biotechnology, zoology, botany, etc. I guess. Therefore, I have chosen a topic that's of interest to all. The title, as you can see, is Evolution of Genes, Form and Function. Any subject that talks about evolution is of interest to all teachers, biology teachers and researchers. And I will be talking to you about what we know about the evolution of genes. Let me start with my first slide. This shows an example of the variety of life on Earth, which basically the plant kingdom and the animal kingdom. As you can see, the variety of life comes in smallest to the biggest forms, the simplest to the complex forms, both among plants and animals. So for a long time, people had no idea. It is only during the 18th century onwards, 17th century onwards, that people started looking that some plants and animals are similar than the others, and they classified according to their similarities. That is the time when taxonomy was born. From the, as you all know, you have studied biology, starting from Leeuwenhoek. The idea of plant taxonomy, classifying based on their close similarity into respective groups or phyla or class or orders, was the basis for us to understanding the structure of the variety of plants and animal kingdom. It was not until Darwin that we had no idea about how life evolved on Earth. As you all know, the life evolved on Earth more than 1 billion years ago, and it was for a long time barren. And with the origin of life, complexity arose over a period of millions of years of evolution. And it took different directions when photosynthesis evolved among the plant kingdom and the complexity of life evolved on land and in water. The animal kingdom, as you know, that the complexity of life varies from single-celled protozoans to the metazoans and ultimately culminating in complex mammals and primates. So the concept of evolution as proposed, conceived, this time gave us an understanding of how life evolved into complexities. And Darwin, for the first time, gave us the most wonderful theory of evolution, where a species evolves from a pre-existing species, and the complexity of evolution can be understood based on the fossil record, and that gave us of understanding of how the complexity of life evolved both in land and on water, and various animals adapted to different surroundings. Therefore, there were modifications and adaptations. This theory gave us a comprehensive understanding about the evolution of life as a consequence of survival, adaptation with modification. This is what we all know, the fundamental. We also further understood that this complexity of life on Earth can be shown that a species or a group of species evolved from their ancestors with very similar structure, but with further modification gave rise to complexity of forms as an adaptation to life on land or water or air. You can see here, if you just take the evolution of limbs, for example, in case of fish, it is the fins. In case of frog or reptile, you will find the limbs. These are the modification of the same structure, evolution of limbs in tetrapod, that is animals that walk on 4 legs or swim or fly. The same thing is modified as a wing in the case of birds. So what actually tells you is it is the same structure that has undergone modification over millions of years for life, either for walking, running, or flying, or swimming. But only thing what we need not understand is how exactly this happened, how the fin became a leg and how leg became a wing. So for a long time, people did not know much about it. It is only with the theory, Mendel's theory of inheritance, where he for the first time showed that there are traits controlled by specific factors, as you all know, which are responsible for transmission of a character to the offspring. So that is the first genetic base of understanding of a trait or a structure for a function. In the case, Mendel actually showed that it was the evolution of particular traits in the pea plants. You all know that. But it was not until Morgan who showed that specific change in a gene is actually responsible for a specific function, as he showed by mutations occurring in the fly Drosophila. That is the day, that is the time when the genetic basis of heredity was propounded. Now we all know genes function, genes control all functions, the genetic basis of life, and now with the structure of DNA that was deduced in 53, and Watson and Crick gave us the structure of DNA, which gave us the whole understanding of the mechanism of how a gene is translated into a protein through RNA. So the central mechanism or the dogma, DNA, RNA protein, was understood in the 60s. So by then, starting from the evolutionary theory to genetic theory, we understood that the basis for the structure and function was genes, and the inheritance is the basis on which a structure and function is transmitted to the next generation. So therefore, in the evolutionary theory, it was propounded that genetic mutation is change of function, which gives rise to variety that is selected by nature during the course of evolution of life on Earth or a species in a given environment. Therefore, the genetic mutations are still considered as the raw materials of evolution. This is a central theory in which we explain evolution. So therefore, this still will not tell us how exactly structure function evolved. So this is going to be the topic of my today's discussion, which has engaged the best minds in the last 3-4 decades. So evidence from evolution of proteins to specific function genes is very well known, the code for them. You all know very well that the cases of genetic disease like gene mutations, like sickle cell anemia, was due to a single gene mutation, which is heritable, which changes the function. And similarly, many heritable diseases by a single gene mutation like captonuria, phenylketonuria, you all know that. But the mutations explain a specific function, gain or loss of function, but it will not tell us how exactly anatomy evolved. It doesn't tell us how hand evolved from fish. It doesn't tell us how the simple brain in the fish evolved into a complex brain of mammals and of primates and man. So simple gene as a concept, as we all know, is a sequence of DNA that is transcribed into a protein, was the basic understanding of our genes. But you all know, majority of the genome, as we now understand after sequencing became very well available everywhere, the sequencing data from the genomes of animals gave us that more than 90% of the genome is non-coding, which means to say the coding sequences constitute only a small part of the genome. Now people understood what is the function of this DNA. Some people it was even understood in the 80s and 90s, even 2000, that there is a lot of junk DNA. Junk in the sense, DNA is meant to be transcribed into an RNA, which then is translated into a protein. But there is a whole lot of DNA which is not transcribed at all. So people believed that it is junk DNA, but nobody knew what its significance. They just believed that it existed as non-functional, and nobody knew what exactly it means. If you compare the genome, now that we have the genome, full genome sequences, after 2000, you know, the first human genome sequence was published. Many species of animals and plants genomes are now available. The sequencing technology has really revolutionized our understanding the genetics of species in terms of genome, because we are now trying to understand how exactly a species or a function or a genetic change occurs, which can be related to normal function or structure or even a disease. So if you just take this example of an evolutionary process of two related species, like we are more related to chimpanzees. As you know, there is 98% sequence homology between humans and sequences. But still this 2%, how does it explain that we are the human beings? This is the biggest question. The sequencing of human genome and the genomes of the animals give us. How do we understand the evolution of a species? Species in the sense, how exactly the differences in the sequence, just 2% what if account for the tremendous differences between the two species. In this case, I'm just giving an example of the monkey and the man, or the ape and the man. How do we explain the anatomical and behavioral differences? If you take the anatomy of a monkey and a man, or a dog and a cat, elephant and a tiger, basic anatomy that serves the survival of the species does not explain how exactly that difference evolved. Are there genes that specifically control anatomy and function? This is the question that actually engaged the minds of people in the 70s and 80s. When genome sequences are not available, there were only examples of mutations that change the functions, but we had absolutely no genetic basis to understand evolution of structure and function. So, in the case of distinction between evolution, anatomy, and physiology, some people even theorize physiological changes as exemplified mutations in specific enzymes or proteins does not explain the evolution of anatomy. So they even people propose there could be specific genes that direct the development or the evolution of anatomy and the physiology. They even called them as morphogenes and physiogenes in 1975. So changes in size, shape, and number of color patterns fundamentally different from change in physiological process as a function of structure and functional differences. It was not until Ono in 1970, I don't know if any of you have read this book. Ono, anybody knows about Ono? Have you read this book? Have you seen this book?
Yes, sir.
Yeah, that's
Yes,
nice to hear somebody. Oh, this is okay. One of our okay. Ono was the first one to come up with a theory that genes evolved by duplication. In 1970, before structure of a gene was known, he had the vision to propose a theory that genes are not statics. Genes change. Genes multiply, same duplicate. And it is the duplication of gene that is responsible for the evolution of genes. This was the first theory that was proposed in 1970 when we hardly knew anything about genes except chromosomal or mutation as the basis of genetic inheritance. Evolution by gene duplication was a revolutionary concept where they could propose that allelic mutations in a gene loci does not account for major changes in evolution because it's a spine mutation, allelic mutation, as you all know. But duplication of genes, particularly the regulatory genes, must have contributed greatly to the evolution of vertebrate. This was a theory proposed by Ono. He first times meant that regulatory genes and their duplication must have contributed greatly to evolution of vertebrates. So let us see what do we know today. This was proposed 50 years ago, more than 50 years ago. Now today we have evidence to show that evolution of gene duplication is indeed a phenomenon that has happened, that continues to happen in the evolution of genetic changes among all species. The duplication as a concept, as you know, there is a gene here which has ancestral function, and this undergoes duplication. You see two copies of the genes. And these two copies of the genes can change in sequence by mutation. One can either become functional or non-functional or specialization, depending on, because as you know, the change or the mutation or duplication is a random phenomenon. And the changes that are selected are based on its utility to the organism, its ability to help the organism survive and reproduce. So the ones that are conserved are the ones that are useful to the organisms to survive, reproduce, and therefore this is the change that is brought about in a single gene here with a duplicated gene. But if it is non-functional or subfunctional, eliminate, either it's eliminated or functional if it is neutral, in selection. So therefore, we now have a concept where the duplicated gene with a mutation acquires the function of a new genetic change as the basis for explaining the evolution of genes. This is the evidence we have now when once we have the genomic sequence data. So, but does anatomy, evolution of anatomy, evolution of structure in terms of the animal kingdom tell us anything about the genetic basis of anatomical changes that has happened during the course of evolution? So, you know, in the 80s, discovery of Hox genes and many other genes gave us a wonderful example that a set of genes, these are not the genes that code for a specific enzyme, these are the genes that code for elements that control gene expression in a whole lot of an organ. So the discovery of Hox gene gave us an idea that it is the potential for coding sequences in the generation of genes by duplication. And this gene duplication also contributed to the evolution of form over a long period of evolution, but not sufficient to account for diversion of the lineage. You know, for example, there are 300 species of beetles, 10,000 species of birds. How does it explain that variety of structural changes among different orders of file of animal kingdom? So one of the wonderful byproducts of the knowledge that we gained from genomes of species, that we were able to explain certain theories that were proposed. In this case, this is an example of the finches. You know, Darwin people, this is a story that goes that Darwin got the adaptation as the basis for speciation when he studied the finches of Galapagos Islands. You all know that. So, but that time, the ancestral species gave rise to 3 different species, depending on the modification that helped the species of the bird to survive, where ecological changes led to changes in food pattern or availability of food resources. You can see there is a finch that adapted for specific feeding habits. So this is warbler finch, woodpecker finch, and ground finch. But this structural change in the beak actually was species characteristic, and we had no idea how to explain. All that was known is that is as an adaptation in the change of beak helped this species to survive, to get to the source of food available, and therefore survive and reproduce. Eventually led to speciation. 3 different species from an ancestral species, which cannot survive if there is ecological change where food becomes scarce, and these species which change their beak form survive. So today we have no data from the genomic sequence that tell us what exactly is the difference that has happened in terms of genomics. It's very clearly, it shows it is the genes that have changed in that has led to species specific changes in the beak. Therefore, mutations that cause variations which are helpful in adaptation ultimately lead to speciation. That's how the availability of the sequence data helped us understand the species evolution at the same time as a function of adaptive evolution. People also looked, taking example of plant species, where they looked at the resistance to specific pests. In this case, I'm not going to delve deep into that. You can go into this. Over a period of time, an ancestral gene that evolved resistant to a particular diverged, where the resistance to the gene that is subjected to different conditions by gene duplication arrived at the same function of resisting, and which is a very similar genetic adaptation of 2 different species, but adapted to disease resistance by genetic mutation and gene duplication. This is an example, also tells us how convergent evolution has happened, which means adaptation to a change in the environment, whether it's disease resistance or food resource availability, can be explained now by the convergent evolution, which also involves gene duplication and mutation. I just gave you the example in plants along with animals. So, what is now understood in the last few decades is that it is not just the coding sequences that account for the evolution of structure function. It is the non-coding sequences which have been responsible for the evolution of structural and functional changes. This was first proposed way back in 1975. Evidence was not forthcoming because we did not have sequence information about the species differences. And now we have both evidence from diverse animals from plants where, your hundreds of species have now been sequenced, which are allowed us to understand the association between gain, loss, or modification of morphological traits in terms of changes in gene regulation during development. I'm talking about gene regulation, which involves non-coding sequences. These examples have been found in fruit flies, plants, and vertebrates. In fruit flies, many structural changes due to mutations of genes or by gene duplication and pigmentation, which are anatomical modification or a morphological traits, are now we are able to dissect the genetic basis of them. Similarly, in case of plants, coloration of flowers, architecture of flowers, and the branching pattern also has given us that this morphological diversification as a basic of evolution of genetic structure in regulatory elements or non-coding sequences. As I told you in the first slide, that the evolution of limb and diversity of axial diversity actually tells us that the evolution of structure for a specific function can be explained through regulatory sequences and the evolution of the regulatory elements in the gene function. Therefore, it is not just the coding sequences as the cause of change, but implicated in regulatory sequences in evolution. That is what was proposed earlier. Now, the present evidence in the genomics tells us that indeed that is what has actually happened during the process of evolution. So let us give you an example how exactly a change in terms of duplication and mutation changes the gene function. If you take a gene here, it's a progenitor gene, an ancestral gene. If there is a mutation in the regulatory element and duplication of gene and a mutation there leads to gene duplication and divergence, you can see here. If you compare these two, there is a mutation in the regulatory element, there is a mutation in one of the duplicated segments, which means you have a gene duplication and divergence. And the same thing, if is useful, if it is selected, it becomes conserved. That is useful to the species. The other mechanism is genetic duplication of the regulatory elements themselves. Here, regulatory elements are not duplicated. Only there is a change in the duplication of the gene. If the regulatory elements are duplicated, you can find, and they are further undergone mutation in the duplicated sequence, you will find expansion of the regulatory elements, which can give rise to isoforms, and which, if they are functionally relevant, are conserved and selected by natural selection. Therefore, gene duplication followed by mutation, expansion of cis regulatory elements, and evolution of new exon splicing as a consequence of duplication. Here is an example. You all know exon splicing. By duplication gives to many exons interspersed by introns, and it is the spiking mechanism that gives rise to one gene giving rise to more than one protein or polypeptide by splicing mechanism. This is one of the things that has happened in regulating or in giving the diversity of gene functions that helps an organism to achieve diversity in function and adaptation. I gave you the example how here is an example, the Drosophila fly, the fruit fly. There is a yellow color. We now know that this yellow color is explained by this specific sequence of the gene, whereas this color as a pigment to confine to the one spot is now explained in terms of the genetic change that has happened, because this is something in the laboratory that happens, and you can also get the species from nature. And here you can see it is the fruit fly where the changes has occurred due to mutation in the duplicated segment, and this has led to the change in morphological trait. These are very close related species, but this is Drosophila melanogastron, this is Drosophila bizarrifus, and you can very clearly explain morphological variation as a consequence of changes in the regulatory sequences. It is a wonderful example to show how regulatory sequence, not the coding sequences, that are responsible for a morphological trait, evolution of a morphological trait, and therefore could be a speciation mechanism. Similarly, people have found in a species of fish that has lost specific genes where it has lost specific elements in its structure, that is in the pelvic fins, which are rudimentary here. And here you can see this is one species which has, due to a loss of a regulator element, has lost its hind limb in the equivalent of fin here, you can see here. So now the closely related species, evolution of loss of function is attributed to the loss of a specific gene here in this particular, where we now have examples to show that specific change in structural elements can be attributed to specific changes in the genes in the regulatory elements. Now this is a wonderful evidence to molecular evidence to show us that indeed the changes in regulatory elements are responsible for changes in trait. I'll give you another example, taking birds. You know, among birds, there are birds that are known for singing. There are birds which are not well-known singers. And as you know, singing requires a specific voice pattern, which is generated through its mouth by a voice box. The voice box that is present squeezes the air through it, and this creates a sound. This sound, if it is called a bird song, is species specific. This is controlled by the brain of a specific pattern. So if you know about the bird song is the language of the birds. They can advertise their presence. They can use this song to attract mates. So this people wanted, that we all knew, but what is the genetic basis of a songbird versus a non-songbird? Is there a genetic difference between a singing bird and a non-singing bird? Just like you have a bird that flies, a bird that doesn't fly. So there are so many functions. People have looked at one specific gene that is responsible for regulation of the function of the song in the brain. This is, I do not have the time to give you that in the songbirds. This is zebrafinch which sings. There is a particular gene that is expressed in the songbirds, whereas it is absent in the non-songbirds. Similarly, in a songbird, this song is normally expressed in the breeding season because it is essentially to attract the mates. And therefore, here is an example where that gene is expressed in the brain only because you need the neural circuits to control the bird song. And this circuit, it has evolved both with the evolution of the voice box as well as neural control or of voice generation. It's a wonderful example to show that a behavioral pattern, which has a structural basis, that involves more than one organ. In this case, the voice box and the brain has evolved into creating a species where it has a specific song compared to another species but does not have a sound. And the song has its own value as an adaptation, as a species characteristic. This is how today, with the modern molecular biology, genomics, we have been able to understand evolution. Therefore, we now can say that contribution of regulatory and coding sequence in anatomical evolution constitute an empirical evolution of traits, gene regulation, and the rate of gene duplication. Regulatory proteins are slowly evolving compared to point mutation, as I told you. The rate of evolution, as you can also, if there is a point mutation, you can find its expression in the next generation, if it is heritable. But if it is a regulatory protein, you can find that a new species evolves and the genetic change or the genetic change is huge. Therefore, compared to the point mutation, it is the mutations regulatory proteins which are very slowly evolving depending on the evolution rate that is occurring in a given environment. Theoretical consideration on pleiotropic mutation is a basis on which we are able to, what I mean by pleiotropic mutations that have multiple effects, diverse effects. So therefore, now there is adequate evidence for us to conclude that the evolution of anatomy occurs primarily through changes in regulatory sequences. That is what we know after the revolution of genomics we have on hand. Getting back to the chimp and human redux, how do we explain? We have now sequences of both chimpanzee as well as humans. And what can we learn to understand how exactly man is different from the chimpanzee in terms of a species? So what describes man as so different from the chimpanzee? We all know that it's brain size, it's a cranium facial morphology, cortical speech that is controlled by the brain, language areas in the brain, and the hand and digital form. Our versatility of using our hands and limbs is very versatile. Accordingly, the brain functions evolved. Those of you know the brain anatomy, that large area of the cortex is devoted to just how the hands are used and the fingers are used. Whereas this is so specific to primate and so wonderful in case of human beings, and the cortex in the brain has evolved to accommodate the functional organization of the brain in order to control the limb movements, which has been a wonderful tool for the species for tool making and using of tools. And in terms of brain size and the language evolution has helped the species to develop languages as a communication. Because as you know, chimpanzees have very limited repertoire of vocalizations. They do not strictly have a language. They only have vocalization, which can communicate to its members, whether it is angry or happy or threat. That's all that requires a speech. But we, as human beings, we have evolved languages. When exactly language we still do not know. But what is the basis of language? What is genetic basis of language? What exactly happened in the brain in the course of primate evolution, which created Homo sapiens? And this language and our ability to use our tools to help us highly successful species. And this is the basis on which we are able to explain only through the evidences we have from genome sequence. Therefore, it's a difficult changes to map changes in genes to traits. But nevertheless, evolution of speech has been understood by evolution of functions of the brain has been understood to evolution of new genes. See, this is something I think many of you, I think most of you are teachers. Here is a concept which is very new, evolution of new genes. What do you mean by a new gene? We all know in biology, a gene can only evolve from the pre-existing gene. A structure can evolve only from pre-existing structure. A species can evolve only from a pre-existing species. Nothing can be created de novo. We all know that, right? So, because a gene or a species or an organism is a copy of itself when it produces offspring, which over a period of time undergoes change. If the change is adaptive, it is saved through natural selection, useful to the organism. Evolution ultimately creates a species with new functions. So this is what we know. But all that we know today is all this change is possible by mutations in a gene whose function we know. But there is a concept that is it possible that the new genes can evolve? That is most wonderful thing that we are now able to think, yes, indeed, it's possible. So this concept is de novo genes. How can a new gene evolve? I, at the beginning, told you that more than 90% of DNA sequence is not transcribed. And earlier people called it junk DNA. Junk DNA means it has no known function, which means it does not transcribe into RNAs and proteins. But what we now know is that it is this non-coding DNA is no longer junk. In fact, it is the source of new genes. It could be source of new functions. We now know, you all know about the microRNAs. A lot of microRNAs are derived from the non-coding sequences. You know about the introns. That is the basis where mutation can occur on any part of DNA. It's not necessary that it occurs only in the coding sequence. Mutations can occur throughout the structure of DNA. It's a random phenomenon. If the mutation is useful, if it is transmissible, if it is selected, it stays with the species and if it controls a trait, it is selected as a species characteristic. Whereas the mutations that occur in the non-transcribable part of DNA, this is a source of a possible new gene. Let me tell you an example. Let's say there is a non-genic DNA. If it is, because there are many introns that are also transcribed, that are replicated or even transcribed, but it is not translated to protein because they didn't have the sequences to form a protein. But in this case, during a mutation, if it acquires a codon, like a stop codon and a start codon, start and stop codons are essential for a gene to be transferred and translated. And these codons, if it is translated into an RNA and then a protein, if this protein has no known function, this gene has no selection. If one of these new mutated genes acquired, because when we say gene, it has to have a open reading frame, it has to have a coding sequence, it has to be translated into a protein. And if by a random mutation causes a change in the sequences, which forms a gene where it is transcribed, but whose function we don't know, because it's a random phenomenon. If the protein has no value, it is not selected. If it happens to be a protein that has in some way has helped the organism, it will become a new gene. This is a theory. This is how possibility of a new gene has been explained. I don't know if you know about a species of fish that was discovered in Arctic seas, where the temperature can go up to minus 40, 50 degrees, where any species cannot survive during the winters. This species occurs in a freezing environment where at that temperature no organisms are, because the cells will be frozen, then it becomes non-functional. And they discovered what is called an antifreeze protein. I don't know if some of you may be aware of it. That means a new protein that has the ability to prevent freezing by interacting with these proteins, which is called antifreeze protein, was discovered. When they found the genetic sequence for it, they found that it has no pre-existing gene. There is a new gene in this particular species which has helped it to survive, adapt in freezing environments, which is called antifreeze gene. And they found the sequence was not known from the genome of the fish among the genes. But to the surprise, they found that, in fact, it resembled and it was derived from the so-called junk DNA. So the so-called junk DNA could be a source of new gene. This is a first example to show that new genes can be evolved. And therefore, we now believe that the non-coding sequences is also a source of evolution of new genes. I can give you another example, which is just published a few weeks ago. in nature, ecology and evolution. One of the questions, as I told you, while we are talking about humans as a species, how distinct it is from a monkey, is the brain size, right? So brain size relative to body weight is the highest among primates in the case of human beings. And the evolution of brain size intimately linked to evolution of an intelligent species like man. So the tremendous ability of the brain to control functions which help the species to become very successful, as in the case of man, as distinct from monkeys, is the brain size, where the cortex has become so big that it completely occupies most of the space. And this brain size evolution we had not understood in terms of the genetic basis. Is there a possibility of a gene responsible for brain size? A recent discovery indicates that they have taken a gene from the known from the database from a monkey, which is present in the nucleus, but it does not have the sequence to go out of the nucleus to be translated. And the same gene is present in humans. Whereas the gene that is present related gene cannot go out of the nucleus to be translated into protein. So they engineered this particular sequence. They put the sequence where it helps RNA to get out of the nucleus, where it is translated into a protein. And since you cannot inject directly, you cannot do such experiments. They do a very simple experiment where now there is a new experimental tool, what is called organoids. You can take dividing embryonic brain tissue and transfer the gene into the brain tissue, grow the brain tissue into an organoid. And they took the monkey gene, put into the human gene with the sequence, put into monkey brain, and it allowed the monkey to grow bigger than the normal size in the organoid culture, which indicates a gene that is not really a gene, but derived from a gene that is not transcribed or translated. If it is put into the monkey, it goes to, this is in vitro. This actually gives us an evidence that the human gene has evolved from a gene that has undergone a modification as a change in the course of evolution. This particular gene helped the organism to acquire functions and therefore as a species. Now evolution of the relatively large and complex brain is explained by the de novo origin of new genes. So this is a very, very new concept, that there is in fact what is called de novo genes. It is a gene that is derived from not a pre-existing gene, but a non-genic DNA. This is an example that has led us to understanding that in fact there is the tremendous knowledge that we have gained from the modern tools available to understand human evolution, evolution of plants and animals, evolution of function, evolution of structure, evolution of adaptation. Therefore, the molecular biology and the genomic sequences are the wonderful tools for us to now understand the process of evolution, which was proposed 2 centuries ago. We still have the tools that are being developed that helps us understand the wonderful theory that gave us the theory of evolution. So you all know this famous sequence: nothing in biology makes sense except in the light of evolution. Look at any structure, any function, any organism, that is a story of evolution that tells us this is a structure that has evolved, which has a function. This was famous quotation by Debzhansky, the famous geneticist and evolutionary biologist, in 1973 in his book. But now we have a statement which is saying that evolution makes something from nothing. What do you mean by that? De novo gene, in fact, it is making something from nothing. We thought the junk DNA was nothing, it has no function, it stays, but now we create something, or the nature creates something from nothing. That means you create gene from a non-genic DNA. This is how the biology evolved over a period of a century. So this is what I wanted to share with you. And if you have any questions, we can devote some time for discussion. Any questions?
Hello, sir.
Yeah.
Yeah.
You please
Rukmini, sir.
tell your name and which college or university you represent. Dr.
I'm
Rukmini
Rukmini, sir,
Reddy,
from GPS Institute of Agriculture Management, Bangalore, sir.
Bangalore. Okay.
It is affiliated to Bangalore University.
Okay.
Sir, in this process of evolution, where does this subspecies, biotype, race, and all which are evolving will be placed, sir?
So what is a species? What is a subspecies? They are only related but different. Race is also, it is the same species which has different characteristics. What is the characteristic? It could be a morphology. It should be anatomy. It should be functional change. Right? For example, you take race is distinct from a species. Species by definition is it can
They should
it
mate
can
with
form
each
a
other.
viable offspring when there is a mating
Viable
within the
offspring.
individuals, right?
Yes,
Whereas
sir.
two different species cannot mate, or if they mate, they do not give you viable offspring. That is a species. The basis is chromosomal compatibility, genetic compatibility. It has to form
Yes.
a viable gamete. So there is what is called hybrid sterility. Closely related species, if they mate, the offspring is hybrid. Okay, that shows the reproductive isolation, genetic isolation. On the other hand, even subspecies, if genetically it is different, it does not produce a viable offspring, it will remain genetically distinct. Whereas race is not genetically isolated. If the two races can mate, give rise to a viable offspring because there is a genetic compatibility. So species and subspecies,
Only structure it will be very
genetically they are compatible, produce a viable offspring. But the
yes.
changes in the race can be a combination of inheritance. Right? So the racial differences in an offspring, viable offspring, can undergo genetic changes to produce a mixed race. Like in human beings, although some people do not think it is a race, we have a Negroid race, you have Mongoloid race, okay, you have Caucasian race.
Caucasian race.
So it is controversial, but if a Negroid person marries a Caucasian person, because this is the same species, it's Homo sapiens. It gives rise to a species, offspring, which is the same species, but gives rise to different morphological traits. It could be skin color, it could be other traits that is specific to a race. So this is how it is ultimately the basis of genetic differences in characters, but it is compatible to produce a viable offspring. Right? So that explains. In fact, very clearly we'll explain. We have explained species, we explained adaptation, we can explain subspecies. In fact, today, if you want to describe a species, you have to give the DNA sequences to confirm its species. Earlier, they were only using chromosome or just morphology.
Only the chromosome.
Today, taxonomic identity has to be confirmed by genetic identity in terms of DNA.
Genetic identity. Yes, sir.
Okay, that's how we identify a species, subspecies, and a race. Okay.
Thank you, sir.
Any other questions?
Morning, sir.
Yeah.
Hello, sir.
Yeah.
Good morning,
Sir.
sir. This is Kavya from Central University of Tamil Nadu.
Okay.
Sir, I have two questions. The first one is many plants and some animals have polyploidy.
Yes.
So that will be like little further than gene duplication, multiple copies of a gene. Is polyploidy some kind of advantage for evolution, an evolutionary advantage for the organisms that have polyploidy?
Yeah, very good question. Polyploidy is the first example before we discovered gene duplication in animals. Polyploidy was known in case of plants. Polyploidy simply means the whole genome is multiplied. You know that. So it has helped people to evolve specific varieties where it gave rise to better yield or better size. Ploidy means it is the genome that itself is duplicated. If it is useful, it has been artificially selected because ploidy doesn't affect the viability of the offspring. Then plants also have asexual reproduction where you can still continue the polyploidy. Whereas polyploidy is different from gene duplication. It is the duplication of the whole chromosome set.
Yes, sir.
Okay. So ploidy is an evolutionary force in plants where polyploidy is successful in situations where it has helped the plants to adapt to surroundings. And if ploidy is not selected by natural selection, it won't be there in the species. So in the extant species, if you see the ploidy, you can know whether ploidy is useful. You can relate it to specific plant structure and function, including reproduction. So ploidy is a very well-known example, an ancient example from the chromosomal days, where the whole genome is duplicated. We also have one extra chromosome. You know, that was the case which people studied earlier, 50 years ago. These were called supernumerary chromosomes. This is an extra chromosome. If it's useful, it is retained. If it is not retained, it is eliminated. And this is the chromosome days. People looked at the chromosomal variation as the basis of species. Today, we have more powerful tools. That is not enough to tell us. We really want to know the genes, regulatory genes, non-transcribable DNA, and its change as a function of adaptation, as a function of species evolution, as a function of diversity, and even many new traits that could be evolved artificially in the laboratory can be created. Now we have CRISPR technology, which it was one of the big developments. It is now a technology. You can create a new gene, you can introduce a new gene, you can modify a gene, cure a disease where possible, introduce a trait. It's much easier in plant than in animals, and it's even used as a gene therapy where they have been able to cure. It was recent few months ago, last year, that they have cured a form of blood cancer by manipulating the gene. There is a mutation in a gene known for a particular blood cancer. You can take out the bone marrow progenitor cells, take out the disease genes, correct the gene, put it back into the bone marrow. It gave rise to healthy bone marrow cells, and the person is cured of the cancer gene or the disease gene. So this is possible in gene therapy, where we can introduce genetic gene editing in a cell. But it's much more easy. In fact, it is now going to be a technology in agriculture. It's already there. Yeah.
Sir, thank you. One more question, sir. Sir, you said there is suddenly prototypes, protogenes emerging.
Yes.
So, sir, does stress have a role in it? Like if an organism is subjected to stress, there are chances of more protogenes evolving?
So try to distinguish what is a stress. Stress is a physiological change in an individual. It's not a species phenomenon. If you are stressed, your physiology gets altered. Stress could be environmental, stress could be psychological. The stress, by definition, induces specific behavioral or physiological changes. In case of humans or even animals, we know stress causes continued. This is acute stress and chronic stress. And the stress, the first thing that happens is in chronic stress, the stress hormone corticosterone gets elevated. Okay, cortisol in case of humans, right? Cortisol and corticosteroid that are responsible, which brings about a lot of negative changes in every organ. Whereas acute stress, this fight and flight is also stress. Immediately you see a snake, you jump. Getting angry every day is a different kind of stress. So that is physiological changes in an organism. It will affect DNA. It will modify DNA. But if it happens in the gametes, we don't know if stress can make a specific change in the gamete. So this change in a gene function can be both coding sequence as well as regulation of gene. Now you have the whole epigenetics coming into question. Food, medicine, environment brings about epigenetic changes. Sequence remains the same. The expression of genes can be affected by epigenetic changes, which are subject to modification by physiological changes, by the environment or hormones, drugs. Okay, and the organism goes through. This is individualistic. It is not a species phenomenon. Okay, you can affect gene expression. People have done some studies, but
Okay,
unless it becomes a change in the gene, it will not be transferred to the next offspring. Right? So you can understand the individual gets changed. In case of cancer, it's a genetic change that is happening in a cancer cell that gives rise to multiply a series of multiplications that give rise to a tumor. That is somatic mutation. Somatic mutation gives rise to only to a specific effect on particular organ. Whereas germinal cell mutation only is responsible for change in the species or a function, right?
yes, sir.
Not a distribution.
Thank you, sir.
Sir, sir, thank you for the wonderful presentation. So I do have a couple of questions. My first thing is that
Tell us, where are you from?
Yeah. So yeah, this is Dr. Anandan and from University of Madras, Chennai.
Chennai, which department?
Yeah, the Department of Genetics actually.
Oh, okay, okay.
Thank you.
Taramani.
Sir, yeah, Taramani.
Oh,
Yeah.
I know Taramani.
Yeah, sir.
I know very well.
Yeah.
Do you know me? I used to come to often. You may not know.
Yeah.
Michael
Okay, sir.
Aruldas is my good friend.
Oh, okay, sir. He is from endocrinology.
Endocrinology,
Yeah.
Oh, okay. Tell me, what's your
Yeah,
question?
sir, so we are speaking about this evolution. Does this evolution always ends up in a positive change or have a positive impact, or it can also have a negative? Which is the major one, sir? The positive changes or the negative?
Let us say, changes always happen randomly. Genetic changes happen randomly. Let us say there is a food shortage in a population. Let's not talk about humans. Let's say given situation, there is a food shortage. Food is present, but it is shortage. Smaller animals which require less food will be able to survive better than larger animals in the same population because they need more food to survive. And if they survive, they reproduce. Their numbers will eventually increase. So that trait that is responsible for the small body size will be retained. It is not that it is useful, it is useful at that particular time. Okay, so changes are random, mutations are random. At any given time, if that change is useful to the organism, it is retained by natural selection. And it is incorporated into the population of the individuals. Those individuals eventually increase in number. You know about Hardy-Weinberg law? If you have studied genetics, you know.
Yes.
A change or a mutation or a frequency of a gene eventually increases in the population if it is retained. Selection occurs only if it is useful. Okay. Any change that is selected only if it's useful. Constantly useful, useless, harmful mutations constantly occur. If a genetic mutation occurs at the embryonic level, if it doesn't allow the embryo to survive, it is eliminated the embryo itself. These are lethal mutations. If it's a mutation that doesn't affect you, these are called neutral mutations. If it's a mutation that helps you to survive, adapt to a given environment, because the organism survives, it reproduces, that genetic change retains and increases in the population. As I told, antifreeze protein. Suddenly one fine day, this species got a mutation, it is able to survive. It is in a place where there is no competition. It can survive there, live and reproduce because of one new gene that has evolved suddenly. Other species cannot survive because they don't have that genetic mutation. So therefore, selection of mutation that occurs randomly based on the contingency of the situation in evolutionary types.
Okay.
We are not talking
Okay.
about today and tomorrow.
Okay, sir, and one more thing, sir. So I think you are the right person to ask this. So we are we are on the verge of creating synthetic organisms.
Oh,
So what would be the impact of these synthetic
yeah,
organisms
that's
on
a good
the
question.
process of
I think
evolution?
as teachers, you should share it with your students. You know, this is one of the most interesting thing that is happening. They call it synthetic biology. That means we now have the tools to change genes, to remove genes, to put new genes. So people have now tried to take out the genes, put synthetic genes. And this synthetic gene, they have done it successfully in bacteria, E. coli. And this retaining the genes that are necessary for reproduction. So they have now successfully created a genome, at least partially, that is copied. Because what you need to know, we know. You need an open reading frame. You need this gene for breaking down the glucose. You need this gene for respiration. And you make the gene and put it. Remove the other genes. And the organism, these are essential genes for survival. This experiment has created a new field where you can manipulate genome in simple organisms. But we can only manipulate genome in case of complex organisms in terms of bringing about specific change. But the whole genome cannot be replaced as of now because we can only manipulate. But synthetic biology is an area where they want to create bugs, organisms to make specific. For example, you can put an insulin gene in a yeast that they have done already. You can produce insulin gene. Insulin is a foreign gene. Similarly, all your vaccines, you can put the gene for spike protein in your vaccine gene, which is injected into you. All that is possible only because your tools of molecular biology, which allow us to put a foreign gene. We can also make a synthetic genome. We have been successful in smaller organisms. So how far we can create a species, we don't know. But in smaller organisms, in viruses and in simpler bacteria, it is possible to put synthetic genome, at least partially.
Thank you, sir. Thank you very much.
Hello, sir.
Yeah.
Sir, I am from Kerala, Sri Kerala, from Kerala Verma College, related to Calicut University.
Okay.
I had a question that
It's a college or a university campus?
I am from college and
Okay.
belongs to Calicut University.
Okay, all right, okay.
That if FOXP2 gene, you explain, no?
Yes.
I'm asking whether any gene therapy regarding FOXP2 genes are available and FOXP2 gene therapy and all are available related to that gene.
What is that? It's not clear to me.
FOXP2 gene,
Yes.
yeah. I'm
Yeah.
asking that any gene therapy related to the same is available.
Related to?
FOXP2.
No, you can actually synthesize a gene now. It's not available. See, these genes are there.
Yeah.
See, here they have actually shown the gene expression as related to specific function. I've shown you that particular example, right? This is FOXP2 gene. It's in birds, right?
The same KE family that is related, case is there, no? KE family.
No. The thing is, this is the regulatory gene, which is responsible for song production. So here, this is actually tells you how exactly related. This is an example for regulatory genes, where it is responsible for song production. We have related to evolution of the species, the song in birds. These related genes in other birds, you see in this gene particularly, this bird, if the gene is there is in a different form. I told you that, okay, let me tell you that example. Okay, see here, there is a gene, this gene has changed, right? Okay. It applies to any gene for that matter. Is that I am not able to. See, if you go to the genomic sequence, similarly song, here you can see what is the difference between them. You can see here in the fish, what is the difference between them. This gene is there, it has undergone change. Similarly, in case of human speech, they have also have a gene. I think I have not put this here. Human speech, they have also studied in monkeys. So there is a pre-existing gene. This is not a new gene for speech. Here in this case, it's for a trait, for song. Song is related to speech. Bird song is the language of the birds. So the song or the voice, because your language is also voice, but it is constructed into words, which has a meaning. That is controlled by specific parts of the brain. There is a circuit, right? So if there is a damage to that part of the brain, you will lose speech. Speech cortex, you know that. It's a very complex neurobiology. But the complex network that is formed is evolved in humans. It is one of the genes, it's not it does not explain everything. The control of neural networks, because if you want to speak, first it comes in your mind, in your brain, then comes into words. People who have lost speech, they can't express in terms of words. You call them dumb. There is a mutation. But it's complex, it's not one gene. Don't try to explain everything by one gene. Okay. The available evidences tells us that there is the evidence to say that specific gene functions in coding, non-coding sequences, how we understand in terms of evolution. Any other question?
Sir,
Yeah.
good morning, sir.
Yeah.
Was a nice presentation. So my name is Dr. Sayyeda. I am from ASF College for Women's Bellary.
The last question, because next speaker has to come. Probably it will be the last.
So
question.
one question, just one question.
Yeah.
Whether mutation affects the evolution process, sir?
Mutation is the raw material of evolution. See what Morgan discovered. He found a white-eye mutant one fine day. They did not know it was a mutation. He mated with the normal eye color Drosophila. He found that white color in the offspring and in F1, F2. Then they discovered that it is a specific change in the trait which can be inherited, which is heritable change. Mutation has been known, right? If you know that definition, you have the answer for the question, right?
Oh, thank you, sir. Thank you.
Yeah, thank you. I think we have no time for further questions. Thank you.
किसान हो,